So, so good evening, one and all. It is really my pleasure, and I think I privilege to participate such a uh, very dynamic interactive session of Science Dialogue series organized by my dear friend Gopalji. Actually, we did PhD together at Center for Molecular Medicine, and I tried to get some photos today, but I didn't get time. Otherwise, I could share some photos with Gopalji of PhD days. Okay, so so the thing that so today I'm going to talk about something which is really a great challenge of this age. That is drug, how to counter drug resistance. So I'm stopping my video because then then I will be audible properly.
Yes, yes, yes.
Yeah. So uh, if you go to the so I'm going to discuss something that's multi-targeting drugs, the magic bullets for drug resistance. So we'll have a critical look towards the mechanism of drug resistance, and this concept what we are coming up is now a very infant stage. So we have hypothesized that, and I will say a lot of stories what going on in our laboratory nowadays. So before that, this is a global scenario of drug resistance. So all over the across the globe of different countries, you'll find uh, antimicrobial drug resistance, include virus, bacteria, fungus, uh, protozoan parasites. So all the microorganisms, they they are under drug pressure because we have been using drug, and they want to survive, and they are developing many ways to counteract the existing drugs. So now as a scientist, as a researcher, our objective is to address the issue with a topmost sincerity and topmost sensitivity. So I have been working in malaria for my PhD days. So just I'm saying some glimpse of antimalarial drug resistance. What is happening all over the globe? And if you see the malaria, malaria basically is not found every country, but it is more concentrated in Africa and some part of Asia, including India. And you might have heard about chloroquine and artemisinin. So all the drugs were evolved, they're discovered, and they worked very effectively. So the global malaria burden suddenly fall, and there's a dream by WHO and many uh, like organization that we can eliminate malaria from the globe, as we have a lot of drugs, and these drugs are working very effectively. They are killing the parasites in the limited period of time. So we can eliminate this medicine, eliminate this disease. So in the meantime, as I mentioned you, there is a survival of fittest principle. So malaria parasites try to develop resistance, and slowly drug resistance observed against different drugs. Now, what happens if drug resistance exists? What is drug resistance? So, what happens basically?
Are
How
you
does it
changing
develop?
slide, Dhaneswar ji?
Yeah, yeah,
So
yeah,
first
yeah.
slide ही दिख रहा है, फर्स्ट स्लाइड ही दिख रहा है. अब
अभी हो
आया
रहा
है,
है.
फोर्थ स्लाइड आ गया.
अच्छा.
ओके
Okay.
ओके ओके.
So I have shown this slide,
Okay,
which is
it was
showing the
not
global
visible to you.
scenario of. Yeah,
Okay.
this global scenario of drug resistance, which include all different kind of microbes.
Yes.
And I have been working on malaria, so my talk could be more focused towards malaria today. So this is the various reports of anti-malaria drug resistance, and what I want, how the resistance develop. Like, so generally we have a defined dose of anti-malaria drugs, and it is given. So few parasites survived due to some mutation having against that in their drug target or maybe in the drug transporter. Then what happens in course of time? So these parasites survived and they proliferate and they become resistant towards the particular drug which is administered. So if this happens, what happens? The recurrence happens. That means a patient is cured by a particular drug. So how do you know that a particular patient is cured? For example, somebody has malaria and he has fever, headache, vomiting, whatever the symptoms related to malaria, and has been drugged, take the medication, and it is cured. All these symptoms gone, and then you will find like after 15 days or 1 month again he suffered from malaria. So that time what happened, that very few number of parasites survived, uh, which are resistant to the administered drug. That population basically uh, increased. So that's why that again the malaria come. At that time, whatever the dose already given before, if you administer that dose, it will not be effective, or you have to change some medicine. Like if you have been, the person has been treated with chloroquine, now he has to given atomycin or some other drugs. So as the uh, resistance already now we have all the per malaria, we don't have a single drug left against which parasite have not resistance. So all the frontline drugs parasite in course of time developed resistance not only against the single drugs, also the drugs in combination. If there is combination therapy is going on to counter drug resistance, and parasite developed resistance against multiple drugs. So then the situation become worse. This is a scenario. This is a picture of what is the impact of artemisinin and partner drug resistance on disease burden in Africa. You can see here. This is scenario three. There is a it's basically correlation between the drug resistance and number of cases. There's a sudden rise of cases happens when this, because antimalarial artemisinin and artemisinin based combination therapy, they were in the front line of antimalarial treatment. So when the resistance happens against those drugs, so in course of time, the number of cases also uh, have been rising. And interesting, you find this is a in the last decade, the number of malaria cases have been increasing. This is a comparative analysis of last 5 years data, that is from 2018 to 2022. You can clearly see the number of cases have been increasing, as well as also if you see the number of death also, 2018, 19, and 2020. So in 20 there is much many deaths due to happens due to malaria, and people have people used to think that okay, so this is corona time is COVID time is going on, that's why people are not getting proper treatment, and that's why death is happening. But see the result of 2021-22, COVID is already gone. That era is over. Still, we have whatever the death cases, death numbers happening. That's more than 2018 and 19. That means something situation is not very easy. It's a very complicated situation now. So now it's an urgent need of for developing something against which parasite uh, cannot develop resistance. So this is a call of a time that we have to develop some antimalarial drugs which is immune to resistance. So before that, try so when you have to fight against resistance, we should know the possible causes of resistance. So there are multiple ways parasites uh, want to survive or try to survive under drug pressure. It may if the drug is targeting one target, so what they do basically, the, and one particular metabolic pathway is getting disturbed, so the parasite try to develop a alternative pathway to survive. So it changes the metabolic pathway. Sometimes they have some stress mechanism, like like drug used to give stress. So they develop some stress mechanism to survive in presence of that drug. And sometimes what happens basically, what are the transporter through which the drugs are coming? So parasite try to develop mutation in that transporter, so that the drug cannot go inside. And sometimes what happens basically, they develop some efflux system, like even if the drug goes inside, the efflux pump basically will take the drug out, will throw the drug out. So and sometimes what happens basically, the drug is targeting certain enzymes or certain proteins which are very crucial for the parasite survival. And the basically the parasite develops some mutations in the exact the amino acids where the drug is supposed to bind. So the drug loses the affinity towards the drug target. So in major cases, this is the mechanism the parasite follow. It develops some mutations in the targets, in the drug target, so that that drug cannot bind to the target and cannot inhibit the function or the related metabolic function. So now, uh, when we analyzed the drug resistance happens due to different antimalarial drugs. You can see there are a lot of SNP, like a lot of single mutations, which are basically in the drug targets or in the transporter, which are basically causing drug resistance. Now, it is not only in case of malaria. If you go for the virus also, these are the report of this hepatitis B virus, cytomegalovirus virus, and also you'll find what the drug targets are there. There are multiple mutations happening in these drug targets, and by which the drug slowly become ineffective. This is a case of SARS-CoV-2 improve, which is a very validated drug target, and you can see that a lot of mutations happens on uh, the drug binding site, substrate binding site, and which doesn't allow the existing drug to bind. So it is basically, um, if you see different. I just discussed about malaria and some virus because we have been working in these two organisms now. But if you see bacteria also, the similar cases happens. Okay, just we thought how to deal with this, how to develop some innovative way for targeting the drug resistance. We try to understand the genetic mechanism of drug resistance. So if a person get malaria, in an average, it, the persons have 10 to the power 9 to 10 to the power 13 parasites in his bloodstream. In the asexual stage, the asexual stage, basically parasite has sexual and asexual stage. Asexual stage basically it's completed in human body. And if a person having the malaria, so he has basically 10 to the power 9 to 10 to the power 13 parasite in his bloodstream in his body. Now, if mutation is a random process, okay, and the mutation according to the genome size, the mutation estimated mutation rate is 1 to 9.7 into 9 mutation per base pair per generation, and it is worth mentioning that parasite replicates very fast. Within 48 hours, one parasite can be 16 to 32 parasites. See the high replication rate, and the more and more DNA replication happens, there is more chance of DNA mutations. So that lead to 1 to 5 per 10 to per 9 asexual. मतलब, that means you have generally you will get 5 mutations in in every day there is a chance of 5 mutations in the genome of the parasite. Okay. So as I discussed before, so drug resistance happens due to spontaneous mutation in the drug transporter and the target enzymes during the asexual reproduction of the parasite in infected person. Because parasites replicate highly, and there's a huge number, rate, huge number of parasites, and the rate of replication is also high. So that provide a very ideal condition for developing more and more mutations, and although that is random, and that random may happens in the drug transporter or drug and target enzymes. So this is the region, and if you and when you studied like different antimalarial drugs, including sulfadoxine, pyrimethamine, methotrexate, clindamycin, on all the cases. Okay, so these things happening like there are single mutations in the drug target or in the transporter, and the drug effectiveness is gone. Now we thought like if mutation happens in one target, for example, if A target, the probability of mutation is PA, the probability of mutation in another target B is PP, and the probability of that mutation in the target C is PC, or like the probability of mutation in the N target is the PN. So probability of mutation in a single target and a multiple target, the multiple target will be low. So the late chance of resistance emergence of drug targeting multiple targets than a single target. So if you can develop a drug which can target multiple targets, the parasite cannot develop resistance in multiple targets together because there is a fitness cost. So what I observed, like as these drug targets are essential for the parasite survival, as if a mutation in the drug target, what is happening? Parasite basically they become less fit in compared to the wild type parasite. But as there is a drug pressure, the wild type parasite will be killed, and these parasites slowly become fit. So anyhow, if there is a mutation happens in a drug target, there is a fitness cost. So the parasite cannot cannot basically generate or cannot induce many mutations in multiple drug target together because due to fitness cost, or that means the parasite will not fit for survival. So then we thought, is it possible that we can develop a drug which can target multiple targets? So it is this hypothesis. Okay, that we are thinking that okay, if you have a drug basically having multiple targets, with their resistance will be low. Then we analyzed the resistance history of different antimalarial drugs. So in that we analyzed. So you can see here, like the first malaria drug is a 16-year quinine. So this resistance happens against quinine in 1910. But actually that time the surveillance is not so much developed. So it is actually exactly not known when the resistance really happened. But it's with the chloroquine, it's introduced in 1945 and mutations observed or noticed in 1957. And then Ogonville, that is 1948, mutation suddenly after 1 year, 1949. Your mefloquine, 1977, it took like 1 decade or 9 years to get resistance. Piperquine, 1978, and just in 2 years there is resistance. Then Atavacan, introduced in 1996, the same year resistance observed. But if you see the artemisinin, it was introduced in 1979, but resistance, which is basically partial, that was noticed in 2008. So basically, it is around a 30 years gap. So then we'll try to understand what's the uniqueness of the artemisinin, against which it took around 30 years to the parasite to develop resistance and which is also partial. So can we get some clue from the artemisinin drug so that we can develop such kind of drug which have a uh, which will be immune to resistance or the resistance happen after a long time. When we try to understand what is happening with the artemisinin, so we found the artemisinin has multiple targets in the parasite. So there's a report. You can see some of the targets is given here, and there's some report like around 20 or 100, 24 targets are there, which are the proteins which are essential. So now they are partially or completely they're inhibited by adenosine. So that from that observation or that from analysis, we came to a conclusion that so multi-targeting drugs may be the magic bullet to counter drug resistance. So now there is a classical method of antimicrobial drug discovery which is going on, and this multi-targeting antimicrobial drug discovery, as I mentioned, it's an infancy stage due to multiple reasons, and I'll share whatever the problems are existing there and how we are trying to resolve it. Initially, uh, our hypothesis was also have been rejected by many funding agencies uh, because there are some notion in the public. Uh, so that this multi-targeting means it is a multi-target, so each may have a lot of off targets. So if you are targeting the the parasite or the microbe, the host will be also affected and it will be a poisonous for the host. So this is a common notion. So due to this also our hypothesis got rejected in many places. But we come up with a strong like strong logic. So that we mentioned in our process proposal. So just I asked many drugs, which are basically the Ayurvedic drugs, okay, which have been used for multiple diseases. So what does it mean? That means it is multiple targets. So we argued if it has been observed or already accepted, and there is a theory of drug repurposing. What is drug repurposing? We have developed a drug for a particular disease, and slowly we found it is effective in other disease, and we have been using that. So if it's already existing since ages, this multi-targeting concept, so why there is a notion that it will off target? So when we come up with that logic and I'll say it like we got then grant for doing this kind of research. So this is a comparison of the classical methodology of antimicrobial drug discovery and the multi-targeting antimicrobial drug discovery. In classical method, what do they do? Basically, they found a new compound focused in possible pharmacodynamics and pharmacokinetics interactions. So from that we know this drug is safe and efficient. Then we basically launch this drug in the market, and people use that drug because that initially it is very effective and very safe also. But in due course of time, there is emergence of new resistance strain and side effects. But in multi-targeting drugs, we liken that targets two or more drug targets of the infecting agent. The potential drug combating infection by multiple essential routes, because we are not trying to kill the parasite in a single route, we are trying to target multiple routes. So it will then the possible this will be the possible magic bullet to counter ever existing challenge of drug resistance. Drug resistance not happens today. As I mentioned, the genetic mechanism is a random mutation and it's a natural process. So now, so what are the benefits? These are some few benefits. Okay, so they improve. So basically, broad spectrum, target multiple pathways simultaneously, reduced mutation impact, synergistic effect, because if the drug is acting in multiple targets, so there will be more synergistic action, like in combination therapy. Then what is how it is different from the combination therapy? Combination therapy is also working in the same principle. Like if you are using two drugs having two different targets, so combination therapy is working the same concept. So how the multi-targeting drugs are more efficient or better than the combination therapy? See, if you want to develop two drugs, like it takes a lot of money, a lot of time to launch a single drug. A launch two drug or three drug together, so it is really it's expensive. And there is another chance. For example, if you develop two different drugs, they are very effective and effective. Can you go? Will it show synergistic effect? May or may not be. It may have drug-drug interaction. And like nowadays, like a lot of people have already they have already different metabolic disorders. So if you take multiple drugs as a combination therapy, it is also challenging the host or challenging the human being. So that's why the multi-targeting drugs is better than the combination therapy because you are using single drug. And see, if you have a multiple drug, there are more side effects also. So the side effects are lower. So overcoming single target resistance, prevent resistance mechanism, and improve treatment outcomes. So we hope the treatment outcome will be better. Okay, so now we thought that multi-targeting approaches were good. Is in the uh, so is it working? The concept whatever we developed. So we try to analyze the market, and we analyze both the science and market. So we found that from 1980 to 2020, you can see here, after 2010, this concept is basically uh, the concept is getting more and more acceptance. So you can see the number of publications it's, they, it has been rising. And we also try to understand what are the FDA approved drugs are in the market. Okay, so how much have in it basically multi-targeting? You can see in from 2015 to 2017, 21% whatever the FDA approved drugs are there, they're multi-targeting nature. So these are very strong logics. We try to convince ourselves that we should try developing multi-targeting drugs for malaria or other viral diseases. So now we started this concept from during COVID time. So um, as I mentioned you, there are a lot of resistance. Like COVID happens due to only mutations in your receptor binding domain of COVID. So then we that time we started working on developing some uh, the multi-targeting drugs by computational method. Then we also tried for monkeypox and then malaria. So we have been in experimental stage in malaria. For COVID and monkeypox, we did whatever is there in the computational approach. Okay. So before that, just I'm going to discuss some of the challenges of multi-targeting drug discovery, which is not in the slide. So see, like developing screening library for a particular target, a single target, is a very costly thing. So you have to develop assay and buy, the chemicals. Like it's very expensive nowadays. Like if you buy a particular a ligand molecule, it costs 5 to 10,000. So if I try to screen like all like compounds, just imagine how much cost. Again, multiple targets. So it is really expensive and it's not. It looks like it's a very impossible task, a very difficult task. I think this is a challenge which that's why people are not much interested to go in that direction. But here, what happens basically, we are implementing computational approach. You know what computer does basically? Whatever we do, whatever we do, computer reduced our cost and also energy, and doing a very fast rate. This that our computer has been doing. So we found that computer is also uh, taking it play a major role in drug discovery. That's a computer aided drug discovery. So keeping this challenge in mind, we used this computational method or computer aided drug discovery approach for developing multi-targeting drugs. So then what happens basically then we started and I will share the story about malaria. So this is the first approach we took developed multi-targeting drugs for malaria. We found so that kinases in other diseases like in cancer in many diseases, kinases are very good targets. They were attractive drug targets. And there are many reasons why we selected plasmodium kinases. We found their significance. We found they are basically uh, very much different from the host. That's why they are druggable. And we selected those targets, which are basically their target-based biochemical assays are available. We selected those targets, which structure is well known. In computer-aided drug discovery, if you find any molecule or any protein which have excellent crystallography, there is a high chance you will get a right inhibitor. So that's why we selected those uh, proteins or those kinases which structure is basically known, the crystal structure is known, and which is only one that's PI4KP, which structure is not known, crystal structure is not known, but we selected it because we found the molecule which is targeting this kinase is in clinical trial phase 2. So that's why considering that point, we include that PI4K in our group. So, and we also try to know like this molecule is very well validated drug target, genetically validated, biochemically validated. So considering all these things, we basically selected these 6 drug targets, these 6 plasmon kinases, which can be which be used for a multi-targeting drug discovery. If you see these 5 kinases, again we use another strong criteria. See, like in malaria, is a very the parasite has a very complex life cycle. It is within human also. It has a liver. There is a life cycle. Extra like hepatic cell like cycle. It is life cycle within the RBC. The erythrocytes life stages. Then it will convert it to gametocytes. Then your mosquito basically take this parasite when drinking blood from the host and then infect another person. Like this is the way like the parasite spread and the malaria disease happens or spread of malaria happens. So what happens basically? We try to understand. We try to also select those kinases which have crucial role in multiple stages of the parasite life cycle. So you can see from this table that. So these kinases were selected which are crucial for the multiple stages. Then what happens basically if you use this, you find an inhibitor which is targeting or hitting these kinases, then it will not cure the parasite, cure the patient. That means if the asexual stages parasites are responsible for the disease, for the symptoms. So if you take the medicine, it hits the asexual stage parasites, then
इसको एक बार
malaria is
स्लाइड शो
getting
मोड
cured.
में एक बार और ट्राई करिए ना धनेश्वर जी।
क्या हो रहा है?
स्लाइड शो मोड में करिए तो थोड़ा सा टेक्स्ट पतला एक बार ट्राई करिए तो इट्स नॉट इंक्रीजिंग है ना, स्लाइड शो मोड नहीं हो रहा है ना। आई डोंट नो व्हाट इज द प्रॉब्लम। ओके प्लीज कंटिन्यू। स्लाइड शो मोड नहीं हो रहा है,
अभी अभी नहीं हो रहा है ना?
चेंज ही नहीं हो रहा, वही है
एक बार फिर से करेंगे वो ये करके,
क्योंकि ये में टेक्स्ट थोड़ा छोटा छोटा है ना तो मे भी विजिबल नहीं होता हो। एनीवे
मैं बोल
कंटिन्यू।
रहा हूँ कि
हाँ
इसको और फिर से थोड़ा ट्राई करेंगे क्या?
हाँ, इसको ये कर लीजिए स्लाइड्स, ये स्क्रीन शेयर को क्लोज करके एंड अगेन, है ना, करिए तो जस्ट ट्राई। हाँ अब फिर से एक बार जो है ना इसको इसको एक बार फिर से बहुत छोटा छोटा दिख रहा था। यस
एक मिनट,
हेलो,
ये आई एम ट्राइंग।
जैसे तो वैसे ही करिए, प्रेजेंट नाउ करिए ना, एक बार देखिए फिर से। यस यस अभी करिए तो इट्स नॉट इंक्रीजिंग, आई डोंट नो, प्लीज कंटिन्यू। चलिए ठीक है, चेंज करके इट्स नॉट स्लाइड शो मोड में नहीं आ रहा है आपका, है ना पूरा। ओके
नहीं
प्लीज कंटिन्यू, है ना।
ओके।
यस
So, so based on these criteria, basically we selected
वहां उस स्लाइड पर
uh,
ले आइए, उस स्लाइड
these
पर।
teacher. हैं?
हाँ,
स्लाइड चेंज नहीं हो रहा है?
नहीं हो रहा है ना?
नहीं 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 हो रहा है सर।
स्लाइड चेंज हो रहा है?
नहीं हो रहा है।
नो नो।
हाँ, हो रहा है अब।
ओके।
हाँ, तो क्या है कि वो फुल स्क्रीन करने पे वो
हाँ,
स्लाइड जो है ना वो ये नहीं हो रहा है, मतलब
ओके ओके ओके, प्लीज
मूव
कंटिन्यू
नहीं हो
करिए।
रहा है.
ठीक।
So,
यस।
so based on this, the criteria, we have basically decided these six kinases as uh, our targets for multi-targeting drug discovery, and we obtained a computational approach, that is a high throughput virtual screening, where you have this happens in multiple stages. I'm not going to in details. Just I am briefly describing like what we did, what we did uh, in this approach. Basically, we select the targets, then we select a library. We selected a cellchem.com library, which is a kinase inhibitor library of 1,801 compounds. Then we prepare the protein. There's a way of uh, protein preparation and ligand preparation. We use a Schrodinger model for this screening. Then we prepared a grid in the target, so that grid means we define the a region on the target where uh, the ligand is supposed to bind, because that is the substrate binding site. And if the ligand bind with the substrate binding site, then the enzyme cannot bind its substrate, and it will be inactive. So we in the docking also we had different stages of filtering using different kind of methodologies. Okay. So STVS, SP, and XP. Then we study two different things. We study docking score and MMGP score. What is docking score? Basically, so you see if the ligand binds in the active site, so it should have a proper orientation, proper conformation. Like that is your basically a structural compatibility, so that it can block the active site. So these things, along with the structural compatibility, how it is binding, and how it is interacting, the docking score is basically analyzed. And this MMGBC score, which is basically analyze the binding affinity, the energy, the free energy. So like the low free energy, the stronger binding. So based on these two parameters, we selected some of the inhibitors, which are basically shown better docking score than the known inhibitors, and better not MMGB score than the known inhibitors. So then we use a quick prop method to understand like whether these molecules are proper, uh, have good your thermodynamic or thermokinetic edema property. Like like if you take the drug, it has to absorb. You have to be distributed throughout the body. It is metabolized, it is excreted. So all these features are very essential for a drug candidate. So if the drug effective, it's not only hit the protein target, it should be also in the host body. It should be properly absorbed, distributed, metabolized, excreted, so that it have no toxicity effect, and it should be effective, and should be alive for that period of time in which it can work properly. So that this quick prop basically analyze these properties of the lead molecules. So we screen those molecules, which basically not only having better docking and MMG score, they are basically fulfill all the uh, criteria for absorption, distribution, metabolism, and excretion. So after that, we try to find out which molecules basically have multiple targets. So we selected those targets, those molecules, which are basically more than two targets. So I'll show that list of the molecules. Then what happens basically, if the molecule having multiple targets, it's binding. So docking squad MMGV score basically, so these analysis basically consider both the ligand and the protein or the target. They are basically fixed. But like the both the protein, like the target as well as the ligand, they are very dynamic when they in the cellular system when they have to work. So in that situation, whether they can interact properly or not. So to that, there's a molecular dynamics simulation we did to that created such a virtual environment, certain extent, having the temperature, pressure, solvent uh, situation, like because they are working in they have to work in the ecosystem. So in these parameters, we also try to understand for 100 nanosecond how these interactions are stable. So based on that, we selected few molecules. Then what we did basically, although everything is happening computer, but really exactly whether they can kill the parasites or not, I don't know. So we treated or we tested against the malaria parasite in culture. From that we found, I'll share the results. So this was target selection,
How
and this
many
is the,
slides left?
yeah, few slides.
Okay, okay, thank you.
So these are the we are coming in another story. Okay, so we find 21 multi-targeting compounds. You can see here. So they are hitting multiple targets, having better docking score and MMGV score than the counter molecules. Then we so these are the control molecules whatever used, and these are the drug-like properties as I mentioned. We analyze ADME properties, and we find these molecules. We selected 21 molecules. They are basically fulfilling all the criteria of having better drugs. Now we study the molecular interactions. The ligand interact with the target with the multiple ways, like making hydrogen bond, hydrophilic interaction, van der Waals interaction, electrostatic interactions. So we analyzed all different kind of interactions happening between the ligand and the target protein. And we analyze the molecular dynamics method, where we have analyzed the RMST and RMSF value. That I'm not going to go to for details of that, those criteria. But overall, you can say that we found in this 100 nanosecond, this molecule, ligand, and the protein, they will have a strong interactions. Then we try to understand whether these molecules are basically similar to the existing antimalarials or they are different. Because say, like if you get some molecules which are similar to the existing antimalarials, already have a resistance against all the frontline drugs. So these drugs will not go for work for a long time. And we observed and we analyzed the how this structurally diverges. So that we do the hierarchical clustering for all the 21 compounds, and we found most of the compounds are very different from the existing, structurally different, the existing antimalarials. So then we analyzed whether these are killing the parasite in the culture or not. If they don't kill, then the story is over. Interestingly, when you took the top three molecules, you can see clearly here all the molecules are killing the parasite in the culture. And one of the compound will get quercetin, which basically having the low IC50 value like 1.846 micromolar. And we have already we have been working in this concept using different drug targets. Currently, there is another story where we found identified some of the compounds which are working in a nanomolar range. So, I think in couple of months we'll find this story will be published. So this story is multi-targeting drugs against the kinases is recently is published, I think, in the molecular diversity. So then we have there's another paper also. This paper is also against different targets where we follow different criteria, and we also publish this work, which basically also for anti-malaria drug discovery. Uh, Again, as I mentioned, that concept we developed during COVID time. So we published these two papers also. We basically absolutely computational because practically we are just entering into malaria field and slowly we'll expand our approach to the virus field. So we worked on COVID also and published two papers. And we doing then monkeypox when come. So we that time we also worked on different targets of monkeypox and we explored some triple targeting drugs against monkeypox virus. So as I mentioned, like we struggle a lot to get grant to work on the concept, and I think this year we get a ICMR grant of 2.28 crore uh, with some multiple institutes where we we got now funding to work on a concept that a novel multi-targeting approach to develop resistant immune antimalarials. So this is our lab. It's and these people have been working most of the time of the day. Vishwajit is a DBT SRF, and Priya and Satyendra they are research scholars, and Varsita is as it usually say JRF. And recently in this project, Dr. Gajendra have joined as a research scientist one. He's basically uh, our group is expanded, and thank you very much to all. Uh, so this is a story like as I've shown a picture of desert where like there is less chance of getting water. So we are like have been establishing this concept and it works. So it will get hope for to counter to combat existing ever existing issues in drug resistance. So thank you very much from thank you very much. So I'm basically working in the Central University Rajasthan. I posted a beautiful picture what the how the university looks during night.
Yes.